Hi, I'm Nancy Washton. I'm a physical chemist by training, and I specialize in solid-state nuclear magnetic resonance of primarily inorganic materials with a focus on metal oxides. I'll be giving a presentation today on the status overview of high-field nuclear magnetics resonance, primarily in the United States and Europe, and I look forward to our in-person virtual interactions in a few weeks. If anyone has any questions or comments, please feel free to drop me an email or give me a call. I'm happy to chat. I'll be turning my video off in the interim so that the focus will be on the slides. So the majority of nuclei in the periodic table are NMR active, as can be seen from the color coding here. But I note that this doesn't imply ease of detectability in a real world setting. Many of these nuclei, especially the quadrupolar nuclei, can be very, very challenging. Isotopic selectivity allows the experimenter to choose a nuclei of interest and interrogate only that nuclei. Typical examples are carbon-13, proton, silicon-29, and phosphorus-31. The information provided is a type of bonding, for example, nonspecific versus covalent, oxidation state, and coordination environment, such as near neighbor identity. Analyte concentration, nuclei identity, the type of experiment, and the magnetic field strength all play a role in experimental success or failure. One method to increase sensitivity is to use probe molecules with nuclei having high NMR abundance as proxies. An example is shown on the right-hand slide, where there is basically an analog of a silicate surface with a fluorinated silane. So you can see the CF3 tail denoted in green, and fluorine-19 is a really nice NMR nuclei because it's 100% abundant. And they also use this in proteins and drugs. So NMR is typically non-destructive, it doesn't perturb the sample, and it can be rigorously quantitative, but you must set up the experiment a priori for rigorous quantitation. Coordination states can be determined, and in the spectrum shown on the right, trigonal and tetrahedral boron have distinctive chemical shifts. In addition, the sharp peaks arise from more crystalline environments. This is especially true when the metal center is in a highly symmetrical environment versus a strained or asymmetrical environment, which leads to broadening of your peak. The insensitivity of NMR requires large sample volumes and may result in long experiment runtimes. In part, these factors can be mitigated by using high-field NMR or dynamic nuclear polarization. In fact, a factor of almost 2 in signal to noise is gained when going from 800 megahertz NMR to 1.2 gigahertz NMR. So here's an example, biological example on a 1.2 gigahertz NMR instrument. So DNAB helicase is a motor protein that catalyzes unwinding double-stranded DNA to single-stranded for replication. This is important for elucidating the global protein structure, especially during the process, as it's critical to understanding the mechanism of action and error occurrence. So color coordinated, purple is 500 megahertz, the lowest field, blue is 850 megahertz, and the red is 1.2 gigahertz. As can be clearly seen from the spectra here, resolution increases as you go up in field. In addition, you can see the expanded regions on the right. The resolution is just phenomenal at 1.2 gigahertz. Now, one other thing to note, going up in field is almost always beneficial, but not always. As can be seen from the one-dimensional spectrum at the top, which is proton carbon-13 cross-polarization, you'll note that around 180 ppm, in fact, the intensity of the peaks goes down as you go up in field. And the reason is as you go up in field, your spectral window width increases and it becomes difficult to decouple protons from the carbon here. And because of that, you get this intense broadening and decrease in intensity of your peak. And so although it's almost always good, there's always a few caveats to be had. So 119 additional resonances were assigned as a result of obtaining these data at 1.2 gigahertz as compared to 850 megahertz. And these peak assignments will further aid in the complete protein structure elucidation. These methods are applicable to any protein where NMR can be conducted on it. So dynamic nuclear polarization provided enhanced signal through coupling a microwave radiation source, the gyrotron, to an NMR spectrometer and a specialized DNP probe. To aid enhancements, the experiments are conducted at 100 to 120 degree Kelvin, and that is the sample temperature. And so I would say that the hardware is significantly more complex than a traditional NMR. There are two means to garner enhancement through DNP, both using radicals. The first is exogenously mediated polarization transfer. A small molecule containing radical is introduced to the sample, typically in an organic solvent or water glycerol mixture into a slurry. Microwaves polarize the radicals, which then transfer their polarization to protons, which are used to then transfer polarization to the X nuclei of interest. The spectrum shows the difference in signal between a traditional NMR spectrum in blue to that of the DNP version with the microwaves on in red. And this is of an aluminum substrate, and you can clearly see the coordination differences, aluminum 4, 5, and 6. And DE, for those of you interested, is direct excitation with the microwaves off. So rather than harnessing the protons, which can be only on the surface of some of these materials to then transfer their polarization to nearby aluminums, the direct excitation is fundamentally looking mostly at the bulk aluminum. And you can see the signal enhancement for using DNP with these introduced radicals. The second method of harnessing radical polarization is to utilize endogenous radicals. In this system, a lithium titanate was doped with manganese 2+. Polarization was transferred directly to oxygen-17, with the spectrum showing the difference between microwaves on versus microwaves off. And as should be noted here, without the microwaves, i.e. doing traditional NMR, there is simply no signal. All it is noise. Now, I must say that this is the most challenging method currently, as the technology is relatively new at Highfield, the DNP technology, and few empirical studies have been conducted trying to utilize endogenous radicals. So here's an example of a biomolecular application at 800 megahertz utilizing DNP. The influenza virus, and this is going to be a mouthful, hemagglutinin stalk long alpha protein helix antigen was investigated. The free version and coupled to virus-like particles. So Highfield DNP provided sensitivity enhancement necessary to detect and elucidate the antigen structure under very dilute conditions of attachment to the virus-like particles. Now, this is important because this is a much better proxy for mimicking in vivo conditions. So if we need to understand the structure of that protein in vivo, this brings us much closer to that than looking at the free protein. So the results are the protein exhibits high structural similarity between the free and bound versions, but the structural differences that do exist were discernible under these experimental conditions. And this was particularly important, in fact, because this goes to vaccines and understanding how to make vaccines that are more effective and what to target. So here's an energy challenge in the more materials and inorganic chemistry space. Identifying more efficient chemical conversions is one of the biggest challenges to a more sustainable future for the planet. Highfield NMR can indeed play a significant role in this area by elucidating structure-function relationships. Aluminas are commonly used as catalyst substrates, sometimes as the catalyst themselves, especially as single atom sites. A key focus is on understanding structural changes to the alumina as a function of catalyst cycling. The electron micrograph shows arrows pointing to the alumina layer on a silica substrate that was put there by atomic layer deposition. These layers are a depth of 1 monolayer to a few nanometers. The NMR spectrum at 1.2 gigahertz, in the plot shown as 23.5 Tesla, shows increased resolution, especially of the octahedral site. In the middle, the molecular model structure will show in dark blue where the authors discerned the aluminum to be in terms of interacting with that underlying silica substrate. So several years ago, there was a report that some of you were part of, and it was a strategic vision for US shared resources. This was put out in 2017. And the big idea was establishing a national consortium of ultra high field NMR and MRI facilities in the United States so that we could maintain not only scientific credibility in this area, but be competitive with the rest of the world. Now, unfortunately, fulfilling the promise of this consortium has not been a priority for US funding agencies over the last 5 years. And this is particularly detrimental. And in 2017, one of the findings is that the lack of access to current commercially available instrumentation is hindering advances in science. And this is even more true today. So commercial high field NMR, although I say around the world, this is almost entirely within the United States and Europe. I was unable to obtain data on high field instrumentation throughout Asia. And so this talk will focus primarily on Europe and the US. So what you should notice right off the bat is that of the systems here that were 1 gigahertz, there were none in the United States, and the majority of them were focused on biomolecular science areas. The instrument at the University of Warwick is the only one that was primarily or only solids, although that surely did not preclude biological solids. So here we have greater than 1 gigahertz. So there's a single 1.1 gigahertz system at St. Jude's. Now, what I would note here is that this is not a university, and so they probably don't have graduate students under their purview. It's certainly not part of their raison d'etre. And so they do have postdoctoral scholars, though, so that helps in the training. The other 3 systems are all in Europe, 1.2 gigahertz systems. And I did find out what the open orders are for these 1.2 gigahertz commercial systems. What you should note is there is only one on the list that's going to be installed in the United States, and that's at Ohio State University. The rest of them are all in Europe, and Germany is by far outdistancing everyone else. So for high field wide bore 800 megahertz DNPs, until this year, all of them were in Europe. The first one in North America, and again, I note this is a commercial instrument. The first one in North America at 800 megahertz is at Pacific Northwest National Lab, and you will see a photo of that system installed in my lab. I'm really thrilled and proud to have that system. It's amazing, although we haven't really hit the ground running yet. And so there is another 800 megahertz system in Bob Griffin's lab, and he's done a lot of development and custom items with that, but it's not commercially available. So US funding sources, and this I hope you find very instructive as I did. The National Science Foundation is the single largest funder of mid-scale instrumentations. They have a call that comes out. The grants are up to $20 million. They, in fact, funded the 1.2 gigahertz system that will be installed at Ohio State for about $17.5 million. Unfortunately, NSF doesn't fund any instrumentation at any of the DOE national labs, which of course breaks my heart. So moving on, National Institutes of Health, I found this to be very surprising. Their maximum instrumentation grant is $2 million. And I did check around because I thought that surely they had something that would allow resourcing for more expensive instrumentation. And there doesn't seem to be at least a standard call that comes out every year. So the Department of Energy funds major items of equipment. The minimum cost there is $2 million, and there doesn't appear to be a maximum cost, which makes sense because they're funding the beamline efforts at the national user facilities. These are typically line items in congressional budgets, and there's a lot of planning and work that goes into getting those authorized. And lastly, there are state and other non-federal sources. So the DNP at Pacific Northwest National Lab was funded actually by the Washington State Department of Commerce. So the Washington State Legislature gave us $8 million to buy equipment because we have a new energy sciences building. And we are deeply appreciative of their support. And we would not have had this DNP without them. There are two nonprofits that I found that fund larger scale instrumentation. One, of course, is the Beckman Foundation. Another is the MJ Murdoch Foundation, although in the last 5 years, the maximum price for instrumentation that they resourced was $1.5 million. So what about the next cohort of magnetic resonance scientists? So I would say within the United States, the National High Magnetic Field Lab is the primary locus for researcher training opportunities. And my assessment of this is based on the fact that they have a specialty in technology and methods development that is rare in the world, I would suggest, as well as having domain-specific scientific endeavors. And they're a user facility. They host hundreds of graduate students and postdoctoral researchers every year. So these students are being exposed to some of the most amazing pieces of equipment in the world and some of the best expertise. And that is truly invaluable. Unfortunately, the US is far behind the EU in training the next generation of scientists in our field. And that is something that we're going to have to come to terms with. Either we can turn this around because we can be resourced, or we will continue to lag until we're simply not competitive any longer. So I am optimistic that with the focus on science and technology with the new administration and with the support of foundations like Beckman, that we can turn this around and we can be the scientific powerhouse that we think of ourselves as and that we have been for a century. So I'm going to leave you with the fact that I love NMR. It's like a puzzle, and it's like piecing together the puzzle in 3 dimensions. And it is just truly fun. And I hope everyone else finds it as fun. And I hope that we can find a way to convey this to students. Just the joy of discovery. And it doesn't even have to be for NMR, just anything in science. The absolute joy of discovery is amazing. And I sincerely hope that we can share that and light the spark in the youngsters' eyes. And I'd like to say thank you for your time.